Hey there, and welcome. Today we are going on a really cool microscopic journey, diving deep inside a flower. We're going to get up close and personal with its reproductive anatomy to figure out the secrets of how they create new life.
You know, we all see their beauty on the outside, right? But have you ever just stopped and wondered about the incredible tiny machinery working deep inside? Well, that's exactly what we're about to uncover. We're going to pull back the curtain on the hidden structures that make it all happen.
Okay, so our first stop on this tour is the anther. The best way to think about this is as the flower's own personal pollen factory. I mean, this is where the pollen grains are churned out in huge numbers, getting all geared up for their big journey.
Now, at the heart of this pollen factory is a structure called the microsporangium. You can basically picture it as a super high security nursery where all the pollen grows up. And believe me, that protection is no joke.
I'm serious, this thing is basically a four-walled fortress. First, you've got the epidermis. That's the outer skin, the first line of defense. Then, just inside that is the endothecium, which not only adds strength, but actually helps crack the whole thing open when it's time to release the pollen. Then you've got these middle layers for even more security. And finally, right at the very core, you have the tapetum. This is the nursery itself, the part that feeds the developing pollen grains everything they need to grow strong.
All right, so we've seen the pollen factory. Now let's switch gears and go see where all that pollen is hoping to land. We're now diving into the ovule, which is, for all intents and purposes, the blueprint for a future seed. This is the female side of the story.
And just like the anther, the ovule is just this amazing piece of natural architecture. It's connected by a stalk called the funicle, which is basically its lifeline. The ovule itself is wrapped up in these protective layers called integuments. They cover almost everything, but they leave this tiny little opening at the tip called the micropyle. And that little doorway is absolutely crucial for the pollen we were just talking about. Inside all that, you find the nucellus, a tissue that's just packed with nutrients. So, buried deep inside all that nutrient-rich tissue, that's where we find the main event. This is what it's all about, the embryo sac. This is where things get really interesting.
So, how does this incredibly important structure even come to be? Well, it doesn't start out as some big, complicated system. In fact, it's this amazing story of multiplication that all begins from just a single solitary cell.
Yep, that's right. This whole intricate structure, the embryo sac, the very beginning of a new seed, all develops from just one cell, which is called the functional megaspore. It's a pretty stunning example of biological efficiency, if you ask me. So how do we get from that one starting point to the final structure? Well, that single cell's nucleus goes through 3 rounds of division. The first division makes 2 nuclei. The second one turns those 2 into 4. And then the third and final division gives us a grand total of 8 nuclei, all floating inside what's about to become the embryo sac.
But after these divisions, the 8 nuclei don't just hang out in a jumbled mess. Oh no, nature is much more organized than that. They sort themselves out perfectly. 3 of them travel to one end, the micropylar end, where that little opening is. 3 travel to the exact opposite end. And the last 2? They head right for the center.
Okay, so now these nuclei start getting organized into actual cells. And this is where we run into a really peculiar and, honestly, a kind of counterintuitive piece of biological math. Let's break this down.
So at that micropylar end, the 3 nuclei form something called the egg apparatus. This is the critical part. It has 1 egg cell and 2 little helper cells. At the opposite end, the other 3 nuclei form what are called antipodal cells. And then, right in the middle, those last 2 nuclei, now we call them polar nuclei, end up sharing 1 single large central cell.
So let's think about this. We have 8 nuclei. And since a nucleus is kind of the brain of a cell, it seems totally logical that we should have 8 cells, right? The math seems pretty simple. But you know, in the wonderfully weird world of biology, the most logical answer isn't always the right one.
And here is the paradox. We have a final count of 8 nuclei. That's totally correct. But we only have 7 cells. And why is that? It's all because of those 2 polar nuclei in the middle. They share the space inside 1 big central cell. So you've got 3 cells at one end, 3 cells at the other, and 1 big cell in the middle, which, you guessed it, adds up to 7.
And that's really the key thing to remember here. When it's all grown up and ready for fertilization, the embryo sac is officially a 7-celled, 8-nucleid structure. It's a unique arrangement that's just fundamental to how flowering plants work.
So, there you have it. We've toured the factory that makes the pollen, and we've explored the intricate blueprint of the ovule that's waiting for it. The stage is perfectly set for nature's next incredible act. But what actually happens when that pollen finally arrives? Well, That's a story for another time.